Hello YouTube, back at it again with one more video for you. This is going to be another installment of the simulation hypothesis line of videos here, part 2. We left off last time here on the game of life. I told you about the walker, this little jumble of pixels basically that looks like an organism or acts like one under the games or under the rules of this game. And I just lined up a bunch of them here, have them, gave them some marching orders, sent them on their way. And last time when I left off the video, I said maybe we'll play some Minecraft next time. And I thought, you know what, the way things are progressing, we're a little bit ready for a few steps beyond that. Let's go straight to the real simulation, which is life. We're going to start here with a Planaria. A Planaria is a type of life form. It's a flatworm, basically. And what this flatworm can do, it has a few cool tricks. If you're this evil scientist, you can cut it in half, and it'll grow a new tail. Now we know of amphibians and reptiles that can grow new tails. This is nothing special. But one thing that this thing can do is it can grow a new tail on the head side and a new head on the tail side. Now this is cool. Multicellular complex organism that virtually lives forever. If you don't mess with it, it can basically live forever. Now we know of other things like
jellyfish. Turritopsis dornii is one of the few animals that can be considered truly immortal. Jellyfish normally go through a life cycle that begins with an egg and then slowly grows to a larva. This attaches to a rock or another solid surface where it continues to grow, becoming a polyp. Once the polyp is big enough, it starts to bud off and the adult jellyfish, known as a Medusa, drifts into the ocean. In the case of the immortal jellyfish, after it reaches its mature adult stage, it can revert back to the polyp stage before growing into an adult once again. Potentially, this can be repeated indefinitely, resulting in a jellyfish that won't die of old age.
But what this thing can do is when you cut off one of its tails, it can grow an entirely new head. Now this is cool and interesting because a lot of people think that most of the information of what an animal is, what it will turn into, is in the DNA, right? As you can see here, slowly the tail slide is growing a new head, some new eyes popping up, an entirely new brain, etc. And eventually you have a working head. Now where did that come from? And how does the tail know to grow a new head? How does the head know to grow a new tail? Why don't you see two-tailed Planaria or two-headed Planaria? They just know. And same with the head side. It cut off its tail and it'll just grow an entirely new tail. Now there's this one concept that was mainly spearheaded from a developmental and synthetic biologist at Tufts University, Michael Levin. He had this concept of bioelectricity. And using bioelectricity, here's a paper here on this, they were able to make these Planaria grow two heads. As you can see here, this growing a new tail. They can make it grow a new head right in this area just by changing its bioelectric code, as they say here. Not changing the DNA or the genetic composition of the animal at all. They didn't touch it. They didn't use CRISPR or anything like that. All they did was change the bioelectric signals, the basically passing of information in between the cells to make it grow a new head. As you can see here, researchers have successfully and permanently rewritten flatworms' regenerative body shape by resetting their internal bioelectric pattern memory, causing even normal appearing flatworms to harbor the code to regenerate as two-headed worms. The findings, published today in Biophysical Journal Cell Press, suggest an alternative to genomic editing for large-scale regenerative control, according to the authors. I highlighted some important parts I thought we should go over, and there's a quote here by Michael Levin himself. With this work, we now know that bioelectric properties can permanently override the default body shape called for by a genome. That regenerative target morphology can be edited to diverge from the current anatomy, and that bioelectric networks can be a control point for investigating cryptic, previously unobservable phenotypes. Now, this is cool because what they're basically saying here is without changing the DNA, we can change large scale properties of biological organisms. How cool is this? Now, here's another little diagram from an interview with Michael Levin and Chris Anderson from TED. And he said with these planaria, these flatworms, you can cut them into over 200 individual pieces. And not only will the head grow a new tail, and not only will the tail grow a new head, but if you cut it somewhere in the middle, it'll grow a new tail and a new head. Now the question is, more importantly, not just how does it know what to grow, but how does it know when to stop? This is the other cool thing about the bioelectric networks within the body. Cells regenerate, obviously.
They're flatworms.
But they know when to stop regenerating.
Actually quite a complex creature. They have a true brain, lots of different organs, and so on. And the amazing thing about these planaria is that they are highly, highly regenerative. So if you cut it into pieces, in fact, over 200 pieces, every piece will rebuild exactly what's needed to make a perfect little worm. So think about that. This is a system where every single piece knows exactly what a correct planarian looks like and builds the right organs in the right places and then stops.
Now this is important. What he's basically saying is once you cut off this little centerpiece, the bioelectric network within the centerpiece knows what the end state looks like. It knows what a full-grown planarium looks like, and it makes the changes necessary to go from this point to this point. Now that's different from the way we typically think about the step-by-step process of the unfolding of DNA slowly coming from a single cell multiplying into the end result, whatever animal you would have. Now let's skip forward here a bit.
Amazingly, there is an electrical gradient, head to tail, that's generated that tells the piece where the heads and the tails go, and in fact, how many heads or tails you're supposed to have. So what we learn to do is to manipulate this electrical gradient. And the important thing is that we don't apply electricity. What we do instead was we turn on and off the little transistors. They're actually ion channel proteins that every cell natively uses to set up this electrical state. And so now we have ways to turn them on and off. And when you do this, one of the things you can do is you can shift that circuit to a state that says, no, build two heads, or in fact, build no heads. And what you're seeing here are real worms that have either two or no heads that result from this, because that electrical map is what the cells are using to decide what to do.
So here you can see these two-headed flatworms that they created in a lab. And it's interesting. He referred to these ion channels as transistors. He refers to this bioelectric network as code to the hardware of the cells. And here you can see a little diagram of this bioelectric network where the red is the head, blue is the tail. So the network itself knows up from down, head from tail, whereas the DNA may not have that information encoded. So here's one more step in the direction to prove that there's something a bit deeper, more fundamental than just purely the DNA that guides an organism through its life. As you can see, they created two-headed worms simply by changing this bioelectric code.
They continue to regenerate as two-headed. Think about this. The pattern memory to which these animals will regenerate after damage has been permanently rewritten. And in fact, we can now write it back and send them back to being one-headed without any genomic editing. So this right here is telling you that the information structure that tells these worms how many heads they're supposed to have is not directly in the genome. It is in this additional bioelectric layer. Probably many other things are as well. And we now have the ability to rewrite it. And that, of course, is the key definition of memory. It has to be stable, long-term stable. and it has to be rewritable. And we're now beginning to crack this morphogenetic code to ask, how is it that these tissues store a map of what to do, and how we can go in and rewrite that map to new outcomes.
So this is very important. They didn't change the genes of these planaria at all yet. Once they have the two-headed worm and they cut that two-headed worm in half, it grows two more heads. Now, how interesting is that? They permanently rewrote the future behavior of these organisms simply by changing the bioelectrical network. How weird is that? Now, going from this simple, flatworm to something a bit more complex.
So when you have tadpoles, for example, you see the kind of thing that most people think is sort of a progressive unrolling of the genome. Specific genes turn on and off, and a tadpole, as it becomes a frog, has to rearrange its face. So the eyes, the nostrils, the jaws, everything has to move. And one way to think about it used to be that, well, you have a sort of hardwired set of movements where all of these things move around, and then you get your frog. But actually,
so what he's talking about here is how you get from a tadpole to a frog.
Organs.
With the old way we used to think about this, we would have the tadpole here. The DNA has a pre-written set of steps, this step by step by step process that you follow to get to the end result, which is the frog. Basically saying the tadpole, the DNA, it knows the DNA itself, knows the end result, and it knows the steps it needs to take to get there. To test this hypothesis, they took a Picasso frog, as they call it, where they intentionally rearranged the face of this tadpole. They moved the eye over here, they misplaced the jaw, they just intentionally moved around the bits of the face. And if we were on this old theory of the step by step by step process, then these steps would take place and you would have a misformed frog. But what they found was something interesting and odd. It's almost as if the bioelectric network within the Picasso-like frog also knew what a correct frog face looked like, tadpole. So it made the steps necessary to rearrange its face to get back on course to look like a normal frog. Now this is cool because what it's basically saying is this coding, for lack of a better term, the information sharing in between the cells knows the end result. And from whatever starting point, whether it's a normal starting point or a misshapen starting point, it does the steps necessary or as needed to get to the end result. Now, this is completely different from the way we typically think about our genome and DNA and how life progresses. And more importantly, what information is stored within the DNA. Now we're saying, well, a lot of this information is actually stored elsewhere.
What you're seeing here is a time-lapse video. For the first time, we are now able to eavesdrop on all of the electrical conversations that the cells are having with each other. So think about this. We're now watching. This is an early frog embryo. This is about 8 hours to 10 hours of development. And the colors are showing you actual electrical states that allows you to see all of the electrical software that's running on the genome-defined cellular hardware. And
so even here, he refers to the cells as the genome-defined hardware. And this green and red here, this is on the borders in between the cells as the electrical software running on this genomic-based hardware. And this is a clear image of this bioelectricity that's being referred to. The information in between the cells. And this network is basically learning and understanding what the end state is going to be and telling the cells more so what to do. I mean, think about it this way. Have you ever wondered how in the world does an embryo in a womb, for instance, how does this part know, okay, now I'm going to start turning into a brain? And how does that part know I'm going to start turning into a nose? And this part, I'm going to start becoming a torso with all the organs inside. How do the stem cells, which branch off into all the more specialized cells in the body, how do they know what to become? Well, the answer is this bioelectrical network, or this light body that coexists with our genomic-based cells, telling them what to do, what to become, knowing the end result.
Cells are basically communicating with each other who's going to be head, who's going to be tail, who's going to be left and right, and make eyes and brain and so on. And so it is this software that allows these living systems to achieve specific goals such as building an embryo or regenerating a limb for animals that do this. And the ability to see these electrical conversations gives us some really remarkable opportunities to target or to rewrite the goals towards which these living systems are operating.
Very, very cool stuff.
Okay, so this is pretty radical. Let me see if I understand this. What you're saying is that when an organism starts to develop, as soon as the cell divides, electrical signals are shared between them. But as you get to, a few hundred cells, that somehow these signals end up forming a kind of, essentially like a computer program, a program that somehow includes all the information needed to tell that organism what its destiny is. Is that the right way to think about it?
Yes, quite.
And he says yes to that. So, but let's not go too far off the ledge here. Let's try to stay in this realm. So let's go to one more part here.
So what you're seeing here is a tadpole with an eye and a gut. And what we've done is we've turned on very specific ion channels. So we basically just manipulated these little electrical transistors that are inside of cells. And we've imposed a state on some of these gut cells that's normally associated with building an eye. And as a result, what the cells do is they build an eye. These eyes are complete. They have optic nerve, lens, all the same retina, all the same stuff that an eye is supposed to have. They can see, by the way, out of these eyes. And what you're seeing here is that by triggering eye building subroutines in the physiological software of the body, you can very, very easily tell it to build a complex organ. Now, this is important for biomedicine because we don't know how to micromanage the construction of an eye. And I think it's going to be a really long time before we can really bottom up build things like eyes or hands and so on. But we don't need to, because the body already knows how to do it. And there are these subroutines that can be triggered by specific electrical patterns that we can find. And this is what we call cracking the bioelectric code. We can make eyes, we can make extra limbs. Here's one of our 5-legged tadpoles. We can make extra hearts. We're starting to crack the code to understand where are the subroutines in this software that we can trigger and build these complex organs long before we actually know how to micromanage the process at the cellular level.
So let's go back a little bit here. When he was showing us this tadpole that had the gut cells that turned into an eye, what he's saying is that the DNA doesn't hold the information. What he's saying is the DNA has a subroutine that knows how to get from, basic stem cells to the end result. And without altering the DNA at all, just changing these, what he calls these transistors, referring to the ion channels in between these cells, we can kickstart those genetic processes. So we can top down turn gut cells into functioning eyes. Michael Levin and his colleagues, they went from, the planaria to the tadpole to the frog, and they went all the way to creating an entirely new type of life form called a Xenobot, using the skin cells of an African clawed frog, or the Xenopus laevis. And these are these little Xenobots. And again, they're created from the skin cells of the African clawed frog. They didn't change the DNA of these skin cells at all. They just use these bioelectrical networks to create entirely new self-replicating organisms. Kind of look like a little Pac-Man there. And this is a simulation of what they thought would take place. And then they would slowly take these individual stem cells you see here. They roll them into a ball and compact them, and eventually, they have the DNA, the genome of skin cells of an African clawed frog, and they turned into replicas are offspring of these Xenobots. Basically little biological robots. Here they say we found Xenobots that can walk, Xenobots that swim, and now in this study we found Xenobots that replicate. Now this is cool because, again, I have to say it one more time. These are skin cells from a frog. So if you left them alone, they're supposed to just be skin cells of a frog. But by not changing the DNA at all and changing this bioelectrical network, we were able to create entirely new organisms from these base cells. Now there are a bunch of different stories that I have here on this. Now, first is a piece by NPR back in 2021, saying scientists say they've witnessed a never-before-seen type of replication in organic robots created in the lab using frog cells. The discovery involves a Xenobot, a simple programmable organism that is created by assembling stem cells in a petri dish. You can think of this like using different cells as building blocks, like you would with Lego or with Minecraft. And here's one creating a tiny little Xenobot baby. And then we have a few more pieces when they went to Xenobot 2. Scientists created a next generation of living robots. They didn't stop at Xenobot 1. They went to Xenobot number 2. Saying these do not require muscle cells to move and even demonstrate the capability of recordable memory. Very cool. Compared to Xenobots 1, in which the millimeter-sized automatons were constructed in a top-down approach by manual placement of tissue and surgical shaping of frog skin and cardiac cells to produce motion. The next version of Xenobots takes the bottom-up approach. The biologists at Tufts took stem cells from embryos of the African frog Xenopus laevis, hence the name Xenobots, and allowed them to self-assemble and grow into spheroids, where some of the cells after a few days differentiated and produced cilia, tiny hair-like projections that move back and forth or rotate in a specific way. So cilia are these hairs. Let's see if we can find a picture of this. So here's a cilia. So like a sperm cell, for instance, will have this one long coiled shaped, spiral shaped tail behind it to help it push forward. But these are cilia, these tiny little hairs, as you can see here, that basically move and allow them to move in one direction or another. They're all over the body, these little cilia. And what they found is, again, they didn't change the DNA at all, but it created its own little cilia, not to be confused with flagella. I forgot the name of that one there. It created their own little cilia to allow them to move around. Saying we are witnessing the remarkable plasticity of cellular collectives, which build a rudimentary new body, in quotes, that is quite distinct from their default, in this case, a frog, despite having a completely normal genome. In a way, the Xenobots are constructed much like a traditional robot, only we use cells and tissue rather than artificial components to build the shape and create predictable behavior. On the biology end, this approach is helping us understand how cells communicate as they interact with one another during development, and how we might better control these interactions. And in this last piece, I thought there were some really cool and interesting snippets from this one. And again, they're doing the same thing. The same team that built the first living robot Xenobots, assembled from frog cells, reported in 2020, have discovered that these computer-designed and hand-assembled organisms can swim out into their petri dish, find single cells, gather hundreds of them together, and assemble baby Xenobots inside their Pac-Man-shaped mouths that a few days later become new Xenobots that look and move just like themselves. And these are these little single cells. How is this possible? If the DNA is the end all and be all of the information stored inside the body, what we're seeing is that is not true. We are working to understand this property, replication. the world and technologies are rapidly changing, it's important for society as a whole that we study and understand how this works. We need to create technological solutions that grow at the same rate as the challenges we face. Replication. Now, this is the big one. If we're to think about, alien life forms or extraterrestrial life forms, why would they be interested in little old us? Maybe it's the same reason we're interested in these tiny little Xenobots. Replication. We talk about the abduction phenomena, Betty and Barney Hill. Maybe replication is at the core of understanding life. How do you go from 2 little robots to 3 robots? How do you go from 2 organisms to 4? Replication. Maybe understanding that one key component of life, how life replicates over time, is the key to it all. And so far, we're under the guise that it's all in the DNA, when really it's not. A large part of it is in the bioelectrical information, or the light body. And in part 3, we're going to dig a little deeper into what we mean by that. But as of now, I hope you enjoyed. And with that, I bid you adieu.